Hi, my name is Emily Leproust. I'm the CEO of Bioscience and one of the three co-founders. I'm very happy to talk to you about writing the future of drug discovery and development today. So first, I'll start by saying that I will be making some forward-looking statement. And I'm super excited because DNA is changing for the world for the better. That's what we do. We write DNA from scratch, and we want to help you solve the many critical challenges that humanity faces. And at Twist, our DNA is used in a wide number of applications. The first one is the sustainable production of chemical. So instead of using oil as a source of carbon, our customers use biomass and fermentation and produce the same chemicals as are produced by oil, but also new chemicals that could not be produced by oil. Our customers are using our DNA to engineer food and, for instance, delivering nitrogen at the root of plants by engineering bacteria instead of using fertilizer. Today we'll talk mostly about therapeutics and the development and discovery of new biological drugs. Our DNA is also used in diagnostics, and last but not least, our DNA is also used in the long-term storage of data. So again, the idea today, the focus today, is to focus on drug discovery and development, where synthetic DNA has a potential to rapidly drive improvement in our ability to treat diseases. So today we're going to hear from experts in different disease and different modalities and different stages of drug discovery and development. And those speakers will highlight how they are addressing the challenges and where the challenge remains and how DNA and the DNA from Twist is helping. So before we go into the details, a quick reminder that today the pharmaceutical industry is facing a real challenge where the cost of developing new drugs is now so high that the rate of return on investment is soon going to be negative. And to reverse this trend, we're going to need new modalities of drugs to increase the number of drugs. And we also will need to increase the efficacy with which we identify new drug targets, and to make sure that we're working on target that will have the highest likelihood of being successful in a very cost-intensive stage of clinical trials. So we need to increase the efficiency with which we identify safe and effective new drug against those targets. And finally, we need to increase the quality and expected commercial value of this drug. And hopefully that's where we can help. So how can Twist help address those challenges? Well, we believe that our platform that miniaturizes the chemistry of writing ACGT on our silicon chip, maximize the throughput. We can make millions of oligos every day, and lower the cost of DNA, which can now unlock a number of applications. So let's start with target identification. The last decade have seen some incredible breakthrough in our ability to discover novel targets for treating disease. And we can't talk about that target identification without talking about CRISPR and how genome-wide CRISPR screening has enabled finding genetic elements that are linked to phenotypes. And thanks to the millions of oligos that Twist makes, now very high quality data can be produced. And here in the example published in 2019 by the Sanger Institute in Cambridge, they've shown that screening with Twist oligo pools of 324 human cancer cells of 30 different types resulted in really high quality data and a number of candidates for downstream cancer therapeutic. But now scientists are able to move beyond just silencing one region by using dual guides. And that creates a bunch of new applications in functional genomic screening. For instance, you can excise promoters, you can excise link RNA, you can excise non-coding elements, or you can actually look at systematic interaction of one gene pair with another gene pair. And so the barrier to entry to be able to do that is actually the length of a dual guide. A dual guide is beyond the ability of normal DNA synthesis. But at Twist, we have addressed that challenge by being able to make 300 base pair oligos. And so that means that now scientists can fit two single guide RNA together. They can fit a Cas12a with a CR-RNA array and basically expand the design space that they have. And so now we're enabling combinatorial screens without having to worry about the downstream cloning issues, without worry about the cost. We can take care of all of it at a very cost-effective manner. And as an example, we are now seeing a lot of great publication in the literature using those dual guide scanning. So now that we've done the target identification, we can go into the drug discovery and development. And we believe that Twist has the potential to accelerate biopharmaceutical and immunotherapy development through rapid antibody optimization and humanization. And that's super important because in 2019, 4 out of the 5 best-selling drugs were biological. So that ability to discover and optimize biological drugs is key. However, it's inherently complex to optimize biological drugs, and the development takes time. And so at Twist, we offer scientists a toolbox to enable them to quickly find new and efficacious leads against targets or even against hard to drug target, which means, and then we give them the means to very quickly optimize drug-like properties for those molecules. And as an example, at Twist we offer two things. Starting at the bottom is our hard to drug target discovery, using our libraries that are inspired by the human repertoire. We've been able to show that we can very effectively pan those libraries and find leads even against hard to drug targets. And then at the top, if you already have an antibody, we're able to optimize that antibody against using the power of our libraries. We can design in the computer massive libraries, synthesize them, and then bind them to find either biobetter or finding molecules that have better drug-like properties. And we've done that at Twist for many different targets. We've done GLP-1R, we've done a number of targets in immuno-oncology like PD-1, TIGIT, CD3, ADORA2A, and we also have done it against COVID-19. On the basis of those great data, we've been able to sign a number of fantastic partners in many different modalities, in many different indications. And if we look at the statistics of what we've achieved at Twist, we've completed 19 different programs, and we still have 43 programs that are active with 31 partners. And again, the indications can be very broad, from cancer to neuro to immuno-oncology, infectious disease. We even do drug discovery and optimization for cats and dogs. And then we have very varied modalities, starting from VHS, antibody, bispecific, ADC, TCR, and more. And as I mentioned, but the root of how we are able to achieve those results is our ability to make amazing libraries. Because we have a silicon chip where we can make millions of oligos at the same time, we can create explicitly millions of genetic elements that have any mutations that you want. And then those elements are super precise. So it's a base by base precision. It's a codon usage precision. We can also precisely do combinatorial assembly of those elements. We can control super precisely the ratio of amino acids. We can control the length of the domains, the length of the CDRs. We can remove restrictions side. We can remove unwanted motifs. We can also pop in that genetic material into many different scaffolds, so that you have combinatorial of CDRs, but also you have combinatorial of scaffold. And then at the end, everything is NGS sequenced so that you know that the quality is there. So again, that is at the root, and that makes sure that you have basically a synthetic library that is human in content. So it's a semi-synthetic library, which is the best of both worlds. And if we contrast those libraries against other libraries like NNS, NSK, or TRIM, we are the only one that can match the natural human repertoire and that can remove the liabilities from the libraries. So now that we've talked about discovery and optimization, we can very briefly talk about on the sequencing side how we can use the Twist tools to stratify clinical trials and to detect a drug response. We have built a series of kits that can sequence and do sequencing of a human sample, but targeting some regions. So you may target, for instance, the genes that, and you can sequence just the gene that the drug will work on to make sure that you can select the patients that will are likely to respond to that antibody. And the main benefit of our NGS assay is that we have lowered the uniformity, we have lowered the bias in the captures, you may see at the top left here. And because of the high quality of our DNA synthesis, we have removed the bias. And what that means is that you need to sequence less to get your answer. And if you only sequence 1 or 2 patients, it doesn't really matter. But if you have to sequence hundreds or thousands of patients, it can very significantly lower the cost of sequencing and lower the cost of the study. So last but not least, to kind of summarize everything that we do, from target identification to target validation to drug discovery to drug development and even to disease stratification, we have the tools at Twist from our genes oligo pools libraries, our NGS kits, and our biopharma products. We have the tools to enable you, help you get to your answer. And so we look forward at Twist to working with you and enabling you to get through those stages and write the future together. Thank you very much.